Greetings, students. So in our last screencast, we really didn't get very far, to be honest, in terms of content. We kind of did this overview talking about how we take the information in DNA and use it to code for a protein, and how proteins are kind of what do all the jobs in the cell. They are what make things happen, whether it's an enzymatic reaction, a cell communication, a transport across the membrane, whatever's happening, proteins are doing it. And DNA sort of controls the cell by directing which proteins were made. And we kind of had this overview diagram where we said that DNA is converted to something called messenger RNA in this process called transcription. And then mRNA is used to make a protein through this process called translation. So before we get into more specifics about transcription and translation, there's one final little piece we need to put in place, and that is we need to talk about the genetic code. So the genetic code is what relates a sequence of 3 mRNA bases to a specific amino acid. Now, you should get a handout in class. It may look like this. There's kind of different ways to structure the genetic code. And here are 2 ways on this diagram. But so it's worth having that out while we go through this. So what is the genetic code? Well, it basically just shows how to interpret the information stored in DNA molecules. So again, DNA, really the magic of DNA is that it is storing information. So just like this little line of gibberish will turn into a website, I think that's a website, I don't know. And this little series of ones and zeros results in some computer display. So too does that information in DNA, that A, C, G, G, T, C, A, that information is meaningful and is used to do useful things like create proteins. So how does it work? Features of this code. Number 1, it is a triplet code. What that means is we break the code down into what are called codons. These are groups of 3 nucleotides that code for one amino acid. So if you look, UUU is the triplet code or the codon that codes for the amino acid phenylalanine. And so we can try this. If I said, what is CAG code for? Well, the way you'd read this, the first base is a C, so you kind of go in this row. The second base, I said CAG, I believe. Second base is an A, so now you're kind of in this column right here. And then the third base is a G, so you'd come down here, CAG, and that codes for glutamine. The code is unambiguous, which means each codon codes for only one amino acid and only one. So if your codon is UUC, that codes for phenylalanine. If it's CCC, it codes for proline. If it's ACC, it's threonine. It's unambiguous. Each codon only codes for one. That said, the code is redundant, which means several codons may code for the same amino acid. And you can see that here. These two, AAU and AAC, they both code for asparagine. So it's redundant. But again, if it's AAU, that's only going to be asparagine. So it's unambiguous. Punctuation is present. You'll notice that there are these what are called stop codons. These are at the end of a protein. In other words, they stop protein production. Thank you, gods of naming. And by the way, you'll see that in this kind of modern era, these topics we're about to move into now has been discovered and figured out more recently. And so the names are no longer these weird Latin-based names. They kind of are named for what they do. So the stop codon stops coding. It's great. There's also a start codon, and the start codon is AUG. That is the one codon that is worth memorizing on here. You'll always have access to this, this genetic code on a test or quiz, but AUG is kind of worth noting because it is always the start of the protein, and AUG always codes for the amino acid methionine. So methionine is always the first amino acid. Sometimes it's chopped off after the protein is built, but it always is the first one to get things going. So AUG codes for methionine, which is always the first one. So there's this punctuation. The final aspect, which is very cool, is that it is almost universal. In other words, the code for a human is the same for the code for a wheat plant, which is the same for a code for like an amoeba, single cell critter, which means it's the same code for bacterium. almost universally, UCC, doesn't matter what species you belong to, that will code for serine. There are a few exceptions. They've found some weird, bizarre single-celled eukaryotes in the organelle DNA of some species. There's some exceptions, but for the most part, it is universal. And this, foreshadowing to a future unit, allows us to do some very bizarre things. For example, there is a gene in deep sea jellyfish that creates bioluminescence. It makes them glow. So scientists thought, hey, what if we took that gene and spliced it into the embryo of a cat? And they did. And lo and behold, it made a glow in the dark cat. They did the same thing. They took the gene that makes the firefly light glow and they put that gene into a tobacco plant and made a glowing tobacco plant. They made a glowing pig. And you may think, what a bizarre, why would you do that? It is a little bizarre. And the reason why they chose this glow in the dark thing was not just because it's awesome and you can now have glow in the dark cats, but because it's a very obvious phenotype. In other words, if you put that gene into the embryo of a cat, you will know if it worked or not. If the cat glows, clearly it worked. If the cat doesn't glow, it didn't work. So it's not just this frivolous, yay, this is fun kind of thing. It was actually an easy phenotype to identify later. So we're going to talk a lot more about that kind of thing. Genetic engineering is what that is called. But we're going to talk more about that in a later unit. Okay, so I think I'll pause there. I'm going to pause there and we'll take the next section in another screencast.